Okay, so we know how you get energy from glucose, but how do plants make glucose? That's the process of photosynthesis, and that's what we're going to look at in this video. So photosynthesis converts solar energy into carbohydrates, in this particular case glucose, but then glucose can polymerize to make glycogen, starch, cellulose, whatever. So basically the equation is 6 carbon dioxides plus 6 waters and sunlight, don't forget that, form a molecule of glucose and 6 oxygens. You do need to know this equation. That's the overall equation for photosynthesis. Notice that it is the opposite of the overall equation for cellular respiration. The only difference, instead of sunlight, for cellular respiration produces ATP. Okay? So let's add a few extra names because we're going to be using them every so often. When we talk about the producers, those are the organisms that produce their own food. We also call them autotrophs because they make their own food. That's what autotroph means, own food. We're talking about plants, algae, cyanobacteria. If they use sunlight as the source of energy, they're called photoautotrophs, meaning they make their own food from sunlight. The consumers, they feed on other organisms that are called heterotrophs because they eat other food. They don't make their own food, they eat the food that somebody else has made. But both autotrophs and heterotrophs are going to use the organic molecules made in photosynthesis. Another important term, pigments. These are molecules that allow photosynthetic organisms to capture the solar energy. Okay? And the most important pigment we're going to be talking about is going to be chlorophyll, the green pigment of leaves. So where does photosynthesis take place? Mostly in the leaves. Actually, any green part of a plant is going to do photosynthesis. But if you look at a tree, pretty much the only green part is the leaves, right? Now you look at smaller plants and the stem could be green. You could have a few parts, but leaves are the major part. A leaf is like a sandwich, okay? You have an epidermis on top and the bottom, and the whole inside is called the mesophyll. And the cells in the mesophyll are the ones that do photosynthesis, that are full of chloroplasts. Now, to do photosynthesis, you have to be able to take in carbon dioxide, so you need openings. The openings are called stomata. You have one represented in here. And those stomata can be controlled by the cell, by the leaf, sorry. They can open and close the stomata. So whenever it's very hot or dry, the plants close the stomata to conserve water. The actual photosynthesis takes place in an organoid that we already know, the chloroplast. Remember, we talked about having a double membrane. Then it has another membrane from the sac that looks like a pancake, like a green pancake that we call a thylakoid. Several thylakoids are stacked up in a grana. The chlorophyll is embedded on the thylakoid membrane, and the space between the grana is called the stroma. So let's move on to the actual photosynthesis. 2 stages. Remember, we have 4 stages with cellular respiration, so this is easier. You have the light-dependent reaction and the Calvin cycle. Once again, you're going to run into different names depending on the book. Some people call this, they don't use it anymore, I don't think, but you may run into it, the dark reaction. Dark reactions. This is a very bad name. The Calvin cycle does not need light, but that doesn't mean that it needs darkness. It doesn't care. So dark reactions is a very poor name because it gives you the idea that it only takes place in the dark. That's why hardly anybody uses it, but like I say, occasionally you run into. I like the name that other people use. They call it the light independent reactions. I like that because you say light dependent means that it needs light. Well, light independent doesn't need light. That is, to me, a very useful name. But some people say, no, we are going to call him Calvin cycle to honor Calvin because he spent all his life studying the Calvin cycle. Okay. But you're going to find light independent and Calvin cycle equally in textbooks. So the light dependent reactions absorbs energy from sunlight. The energy from the sunlight is used to make ATP and NADPH. Notice in the cellular respiration, we use NADH. Okay? In photosynthesis, you use NADPH. These are different molecules. That P actually stands for phosphate, but I tell the students P should remind you that's photosynthesis, that's plants. Both of them begin with P. So it's easy to remember. Okay? The light-dependent reactions release oxygen. We're going to see now where it comes from, and they take place in the thylakoid. Now, once you have ATP and NADPH, the Calvin cycle uses it to convert carbon dioxide into sugar molecules, into glucose. And that takes place in the stroma, outside of the thylakoid. Okay, so we're going to study each one into a little bit more of detail, not too much. I don't mean to scare you. We're talking about light. So what is light? Light is actually a form of electromagnetic radiation, and the electromagnetic spectrum is really big. But visible light is just a very narrow part between 400 nanometers and 700 nanometers in wavelength. Okay, so on one end you have violet light that has the short wavelength but very high energy. That's why next to that will be UV light. UV light is not visible. If you have a UV lamp and you say, I can see the UV light, no. What it is it gets a little bit of violet, and that's what you're seeing. You do not see, humans don't see UV light. But UV light can cause damage to DNA. That's why if you're very fond of sunbathing, you may one day develop skin cancer. Okay? On the opposite end, you have red light that has low energy. So under this, you have the infrared. That's what you use as heat lamps. Okay? But the visible light goes between the violet and the red. Now, different pigments absorb different wavelengths. Okay? So you see there in green chlorophylls, and mostly chlorophyll A and B, these are the more common ones. You see carotenoids. There's actually other pigments, but they're less abundant, because the idea is to have pigments that absorb every single wavelength of the light, so nothing is wasted. Okay? Notice also that chlorophylls do not absorb in this narrow range in here that corresponds with the green light. We'll come back to that, okay? Just remember. But you have many pigments, chlorophyll A, chlorophyll B, carotenoids, and others that they're less abundant to absorb every single wavelength of the visible light. So why are plants green? This is something that confuses students. Oh no, they don't know they're confused. They say, oh, that's because it absorbs the green light. Nope. No, it doesn't. Because if it absorbs the green light, you didn't see it. Actually, chlorophylls absorb everything but green, so the green is reflected, and that's what you see. It goes to your eye. Oh, I see green. Remember, the green part of the visible light is not absorbed by the chlorophylls, so you see green. Now, there are other pigments, which means that you have other colors, but because chlorophyll is the most abundant, it kind of covers everything else. Now, when the fall comes, chlorophyll starts breaking down, it stops being synthesized, so the leaves start losing chlorophyll, and now you can see there are pigments because you don't have the chlorophyll covering them. So that's one of my favorite exam questions. What causes the leaves to change color in the fall? It's not that new pigments are formed, it's that you lose the chlorophyll, so you can see there are pigments that were already there. You just didn't see them. So to talk about the light reaction, we need to talk about photosystems. Photosystems are just clumps of pigments in the thylakoid membrane that they absorb sunlight. And I'm talking about forms because sunlight is, most people think, oh, that's a radiation. Well, yes, but it's packaged. Okay? You can have only certain amounts. You can have 1 amount or 2 amounts or 3 amounts, and we call those amounts forms. They're packages of light. We're not going to waste too much time discussing that. Okay? Again, this is not chemistry. There's 2 photosystems, 1 and 2, and they were named that as they were discovered. So the one that was discovered first was called photosystem 1, the one that was discovered second was called photosystem 2, and unfortunately for the students, photosynthesis begins with photosystem 2. Okay? It was just the way it was discovered, that's the way they were named, no problem. So anyhow, you had the photosystem 2 represented here in green. When a photon of light hits the chlorophyll, an electron gets excited. When we talk about an electron getting excited, it means it gets a lot of energy. Goes up in energy and it breaks free. We'll see what happens to that electron later on. The problem now is that this chlorophyll doesn't have that electron, so if another photon hits in there, nothing will happen. That chlorophyll needs to get an electron back. Where does it get it back? From the breakdown of water, the splitting of water. So when water breaks down, it forms oxygen, it forms hydrogen, and it forms an electron that goes to the chlorophyll to replace the electron that has been lost. So now another photon can hit, you lose another electron, the water replaces it, and the process continues. That's why it's so important for plants to have water. If they don't have water, they cannot do photosynthesis because they cannot replace this electron. Now, what happens with the excited electron? It's going to go into an electron transport chain that's very similar to the one that we saw in the mitochondria. Okay, it's again a bucket brigade where one particular molecule picks up an electron, passes it to another molecule. The energy release as you pass it pumps electrons. This time pumps the electrons from the inside of the thylakoid, from the outside of the thylakoid to the inside. So this is the stroma, this is the thylakoid, so the prons start accumulating inside of the thylakoid. Obviously, later on, there's going to be an ATP synthase, so the prons are going to move identical to the one you find in the mitochondria, produces ATP. The big difference is you don't have oxygen as a final electron acceptor. That electron, when it gets to the end, it goes to the photosystem 1, and the chlorophyll in the photosystem 1 accepts the electron and uses it to make NADPH. Why? Because light is going to hit photosystem 1, an electron gets excited, that electron is used to make NADPH, the chlorophyll needs to get an electron back, it takes the one that was coming through the electron transport chain. So you have 2 photosystems, both of them lose an electron. Photosystem 1 gets an electron back from the electron transport chain. Photosystem 2 gets the electron back from the breakdown of water. This is a brief summary. Unfortunately, when I copied this, it didn't show everything in there, but this is supposed to be water. So this is the photosystem 2. It loses an electron, it gets it back from the water. Now you go through the electron transport chains, and protons are pumped inside the thylakoid. They're going to go through the ATP synthase to make ATP. The final electron acceptor is the chlorophyll of photosystem 1, because when light hits, photosystem 1 loses an electron, so it's been replaced with this electron, and now that electron is used to make NADPH, and the ATP and NADPH is going to go to the Calvin cycle to produce glucose. Takes carbon dioxide, makes glucose. So this is a very nice summary of what's going on in the cell. Now, the Calvin cycle is a cycle, meaning it's a series of reactions. Okay, it's not one reaction, but you begin with carbon dioxide from the atmosphere and that with glucose. Where do you get the energy for all of this? From the ATP and NADPH that were formed in the light reactions. We can divide the cycle into three stages: carbon fixation, reduction, and the regeneration of ribulose biphosphate. Like we said with the Krebs cycle, the first molecule in the cycle to react will have to be regenerated at the end so the cycle can continue. So the first stage, you have a 5-carbon molecule called ribulose biphosphate binds to carbon dioxide, and that reaction is catalyzed by an enzyme called RuBisCo. This is the only enzyme I'm going to mention by name. This is very important because it's the enzyme that we use to control the Calvin cycle. Basically, RuBisCo is going to be inhibited by oxygen, and that will become very important later on. Okay, but anyhow, you have a 5-carbon molecule, picks up one more carbon, so you form a 6-carbon molecule that breaks almost instantly into 2 3-carbon molecules that we call 3-PGA. And I'm sorry, these abbreviations, you're going to say they're all very similar. Yeah, but still easier than the full names, I promise. In the second stage, the NADPH and ATP that we made in the light reactions are used to convert the 3-PGA into G3P. Now, if you go 3 times through the Calvin cycle, you form 6 G3P. Remember, you had 2 3-PGAs, so if you had 2 of these ones, you form 2. If you do this across 3 times, you end up with 6. You need 6 because 5 are going to be used to regenerate the ribulose biphosphate. How does that work? Well, we said this one is 3 carbons. So 5 times 3, that's 15 carbons. If we go 3 times to the cycle, you have 3 ribulose, and we say have 5 carbons. So you can see you have the same number of carbons you're just rearranging. If you use 5 out of 6, it leaves 1 G3P left that comes out. But that cannot make a glucose molecule because that's 3 carbons. If you have another G3P, then you make a molecule of glucose. Because remember, glucose has 6 carbons. So that means that it takes 3 turns to make 1 G3P. You need 2 of them. So altogether, you need 6 turns of the Calvin cycle to make 1 molecule of glucose. Here you have a brief summary. This is 3 turns. So you begin with 3 ribulose biphosphate. That takes 3 carbon dioxides. It makes 3 PGAs that eventually are going to form 6 G3Ps. No, 6, sorry, in there. 5 go back to regenerate ribulose, 1 comes out, and when you repeat this a second time, then you can make 1 glucose. This tells you how much ATP, how much NADPH you're using and all that, without getting to the details of the chemical reactions. What factors affect photosynthesis? One is the amount of water. Of course, we said you need water to split it to regenerate the electrons in the photosystem too. So plants in the desert, they're going to have a very waxy coil to minimize the water loss. And we're going to see that plants that are in any hot conditions, they're going to make some adjustments to photosynthesis to protect their water. You do need water to have photosynthesis. Light intensity, obviously, the more photons you have, the more photosynthesis up to a point. Okay, when every single chlorophyll molecule is being used, you could keep on adding photons and you can't speed up photosynthesis anymore. Finally, temperature. These enzymes that are involved in here have an optimal temperature between 0 and 35 degrees, which is actually a big range. 0 degrees Celsius is freezing, 35 is hot, you're getting to the low mid-90s or something like that. So most plants have a very wide range at which they can do photosynthesis, but some plants are going to get higher, and they're going to have to adapt. So plants are adapted to the light, the rainfall, and the temperature of the environment. The photosynthesis we just studied is called C3, because if you remember, you make a 3-carbon molecule. But this is the photosynthesis you use under moderate conditions. You get enough water, the temperature between 0 and 35 degrees Celsius, blah, blah. When the water is very hot or dry, that's supposed to mean, and you get C4, and we're going to study that. When you're in the desert, very hot during the day, cold at night, you have a different kind of photosynthesis called CAM photosynthesis. So C3, we already seen it. It's exactly what you expect. Carbon dioxide comes in, you make ATP and NADPH, so carbon dioxide enters the Calvin cycle, and you eventually produce glucose. As long as you have the right temperature, you have enough water, everything works out just fine. And remember, this takes place in the mesophyll cells. Now, what happens when it's hot and dry? It's critical to prevent water loss. So you do that by closing the stomata. If you close the stomata, water cannot evaporate. But that stops carbon dioxide from coming in. And worse, it allows oxygen from building up, and remember we said that will inhibit RuBisCo. So some plants like corn grows during the summer, right? It's doing photosynthesis at temperatures higher than normal, and many times under dry conditions. Okay? So what plants like corn do is C4 photosynthesis, called that because you form a compound with 4 carbons. Notice that there's 2 different kinds of cells, and we're not going to get into details because we're going to be studying plants in the second semester, but you have mesophyll cells and bundle seed cells. All of this takes place still during the day. Carbon dioxide is absorbed, makes that 4-carbon compound, and then the plant can afford to close the stomata because the carbon is going to be transferred to the other cells, and the Calvin cycle takes place in the bundle seed cells, and that's where glucose is formed. So that way you're shielding these cells from accumulation of oxygen, because the light reactions take place in here. This is where oxygen accumulates. Okay, it can accumulate as much as it needs to, because ribulose by RuBisCo is on a different cell, so it's not hurt. The plant can afford to close temporarily that stomata, and the Calvin cycle continues in a different cell. There's a physical separation between the light and the light reactions and the Calvin cycle that allows the plant to open or close the stomata depending on the temperature. The desert is different because it's always going to be hot during the day. There's no hope of it cooling. It's always going to be cool at night. So at night, the plants open the stomata. They take carbon dioxide and they make a 4. Notice this is a mesophyll cell, by the way. There's only one cell. But now this 4-carbon molecule is going to be stored in a vacuum. Why? Because the cell does not want to do Calvin cycle or try to do Calvin cycle, because when the sun comes up and it gets hot, the stomata is going to close. Oxygen is going to accumulate. So basically, the Calvin cycle is going to take place during the day. Stomata can be closed at that point. There's no harm. When the nighttime comes, the stomata will open in there. It has a stored carbon dioxide. It has NADPH and ATP from the light reaction. So during the day, the plant can do the Calvin cycle. There is separation between the light reactions and the Calvin cycle, but it's not a physical separation. It's only one cell. It's a temporal separation, night versus day. Either way, plants adapt to the environment. The last thing I want to mention is that the reaction of photosynthesis and cellular respiration are going to share the reactants and products. Think about it. A photosynthesis, you take in carbon dioxide, and what? You make glucose. Then in cellular respiration, you take the glucose and you make what? Carbon dioxide and water. So you recycle these molecules continuously. What changes? Well, in photosynthesis, you get sunlight. In cellular respiration, you produce ATP. So I like to call this the energy laundering. This is my term, okay? You won't find it in any biology book, but I call it energy laundering because what you're doing is changing sunlight into ATP. So yes, I know, this is overwhelming, this is too much. That's why I have that chart that I told you before with cellular respiration and photosynthesis. Again, you're going to divide into different stages, write down in what part of the cell each stage takes place, what do you begin with, what do you end up with, any special requirements. We summarize cellular respiration and photosynthesis in one easy to memorize worksheet or table, whatever you want to call it. But especially I like it because it shows you parallel the two processes, and you're going to see similarities that you cannot see as you go through the chapter, and those similarities will make the memorization you do need to do much easier. You, rather than memorize everything, you will be able to predict what's going to happen next, and that's a big advantage. Okay, so again, in the video description you have a link to my wife's account. Make an appointment and I can give you the worksheet.